Okay, everyone. Thanks very much. Thanks to the organizer. This is Xiong from Nanjing Tech University, China. It's a great honor to be here to share my work with all of you. But today, because of the camera, I cannot share my face with all you guys. Anyway, today I will make a brief introduction of our recent work on salmon species identification using loop-mediated isothermal amplification method. Briefly, I will first explain why we choose salmon, particularly Atlantic salmon, as a research target, and then two methodologies, including the visual lamp and the floss lamp identification method, were introduced for rapid identification of Atlantic salmon. Part 1, research background. Generally, salmon is represent one of the most economically important fish species worldwide, with the common species such as Chinook salmon, trump salmon, pink salmon, mussel salmon, sock salmon, rainbow trout, and Atlantic salmon. Among them, Atlantic salmon is one of the most appreciated and not as widely affordable in China, and has been considered as an excellent source of quality protein and essential macronutrients, crucial for the balanced nutrition and optimal health. Moreover, this species is generally consumed as processed product, such as fresh sushi, sliced on ice, smoky fillet, dry dressed fillet, and even powder to add to baby food. Moment. While in China, salmon was translated as Sanwenyu, according to our previous investigation. This word is most commonly used on behalf of Atlantic salmon on the market. While due to the absence of harmonization around the definition of the word Sanwenyu, over time, Chinese population has begun to increasingly use this term for other salmon species, leading to great costs. Particularly, the recent investigation about COVID-19 contamination in imported Atlantic salmon products has led to great public panic. Moreover, Atlantic salmon is generally considered as an expensive species, with the price reaching 180 RMB per kilogram, much higher than other species, or especially other salmonid species, such as rainbow trout, only 80 RMB per kilogram. However, due to the destruction of the morphological traits during processing, it is quite challenging to make species identification according to the residual characteristics, paving the way for species adulteration. For instance, previous studies have revealed the species substitution of low-cost rainbow trout for high-priced Atlantic salmon for fillet products in South Africa. Moreover, the adulterated species for Atlantic salmon were further expanded to trump salmon and mussel salmon in a recent investigation in Madrid, and to sock salmon in another survey conducted in Canada. While in China, we also made an investigation and revealed more than 8 or 9 species from salmon products. To protect consumers' rights and ensure the public health, rapid and reliable methods to identify Atlantic salmon in processed food products are highly required. Generally, fish species authentication can be achieved using several analytical methods, with DNA-based methods as the most widely used one. Since the development of PCR, a wide variety of diagnostic methods have been established, such as DNA barcoding, real-time PCR, and multiplex PCR. While nevertheless, a significant inconvenience with all these PCR-based techniques has been the necessity to include the expensive instruments and the specialized experts for data analysis. Instead, over the last years, isothermal amplification methods has received increasing attention, with loop-mediated isothermal amplification, also we call LAMP, as the most common one, as we can see from the figure. LAMP actually was first introduced by Notomy in 2020. In 2000, relying on a single enzyme with displacement activity, this methodology uses 4 specially designed primers that recognize 6 regions on the target sequences, producing extremely high specificity and sensitivity. Moreover, the amplification can be made in about 1 hour under a constant temperature, generally 60 to 65 degrees, without initial denaturation step and any temperature change throughout the reaction. Positive samples can quickly be made visible to the naked eyes according to the rising turbidity or color change, or to be checked using agarose GI electrophoresis. Due to the advantage of rapid detection, sample operation, high sensitivity, LAMP has been widely used in clinical diagnostics and pathogen detection. In recent years, several studies have also utilized LAMP for food authenticity purposes. Here we first explain the visual LAMP measure for Atlantic salmon identification. Since the inhibiting dye possess specific molecular structure that allow them to bind selectively to diverse strand DNA. For successful LAMP amplification, as we can see from the figure here, color change can be obtained by adding fluorescent dyes. And a number of fluorescent dyes have been used for qualitative monitoring, such as sap-green. And also we use sap-green in the present study. To establish the LAMP assay for Atlantic salmon, also to ensure the highly efficient LAMP assay, the designing of species-specific primer set is quite important. Here, after analyzing a large number of the mitochondrial DNA sequences, short fragments of the cytochrome B gene were selected as molecular marker for LAMP assay in the present study. With regards to the newly designed primers, in this study, several mismatches against the non-target species were observed at 3 ends of F3 primer and 5 ends of B1C primer. As we can see from here, the primer alignment, explaining the high specificity of the primer set on Atlantic salmon. Before the analysis, the real-time fluorescence LAMP assay has to be optimized. To this end, gradient concentrations of outer primers, inner primers, magnesium, buffer, DNTP, and sap-green were prepared. An optimal concentration for each constituent was defined as the concentration that resulted in the shortest CT values, as we can see from the figure. The results highlighted the highest efficiency of the real-time fluorescence LAMP assay by adding 10 per buffer of 2.5 microliter, inner primer of 1.2 micromole, outer primers of 0.4 micromole, magnesium of 4 millimole, DNTP mix of 1.2 millimole and 0.2 per sap-green. After the optimization of the reaction system, the analytical specificity of the real-time fluorescence LAMP assay was tested using 5 closely related Salmonid species and other 6 common species and other species from different orders. As expected, the typical amplification curve were only obtained using in the test using Atlantic salmon as a template. Sensitivity of the LAMP assay was verified using 30 commercial fish products. Sensitivity of the LAMP assay was assessed by 0.10-fold dilution of the Atlantic salmon DNA from 50 nanogram to 0.5 fetagram and the lowest amount of DNA can yield a detectable signal was taken as sensitivity. As we can see from here, the minimum amount of the detected DNA reached 5 petagram. Practical applicability of the LAMP assay was verified using 30 commercially finished products. The results showed the fluorescence curve could be observed in the test samples SAR1 to 5 and SAR29, about 20% of all the samples. The results were cross-confirmed using PCR sequencing, and the neighbor joining tree clearly revealed that 4 samples, SAR1 to 5 and SAR29, were actually Atlantic salmon, and their sequences clustered with that of authentic samples consistent with the LAMP results. Finally, the detection of LAMP of Atlantic salmon with the LAMP developed in this study take only 105 minutes, faster than the PCR assays. Despite all this, several disadvantages with visual LAMP have been revealed. Firstly, is the carryover contamination during the post-addition of dyes, and also using these non-specific fluorescent dyes may increase the likelihood of false positive results. To this end, we target specific detection of LAMP applicants can be achieved through the use of target-specific probes or modified primers as biorecognition elements. Of these sequence-specific methods, a much simpler, non-Fourier-based approach called fluorescence of loop primer upon self-decreasing lamp, or we say floss lamp, was developed, whereby the fluorescent quencher or quenchers, in the absence of a dedicated quencher, has recently proved its versatility and adaptability. Specifically, a primer, in the present study, we use a loop primer, as we can see here, the red one. It's modified with a fluorophore. The fluorophore attached to the loop primer, we call LBFM, is self-quenchered in abundant state, possibly due to the interaction with the surrounding nuclear base, especially here and here. After binding to the dumbbell-shaped DNA target specifically, the found fluorophore is dequenchered, resulting the fluorescence development. This is the mechanism of the floss lamp. Actually, in the very beginning, four loop primers were designed. Here we can see LF1, LB1, LB2, LB6 were designed. As we can see here, LF1, LB2, LB6 produced an early amplification signal than LB1. Although no significant differences with CT values were observed for LF1, LF2, LB2, LB6, a reduced specificity was revealed for LF1 and LB1, as we can see from figure 2. Therefore, only LB6 was finally identified as a candidate primer to design the self-quenching fluorescence probe. Moreover, we also compared the loop primer and inner primer. Here we use BIP primer for probe designing. As we can see from figure 3, where the form labeled BIP was used to amplify Atlantic salmon target DNA, only weak signal could be detected for all the trials, neither the LB1, LB2, LB6, while the LB6 form always produces the earliest amplification. Here you can see this curve. This is F. We use the LB6 form, and also the signal is stronger and stronger than the intercalating dyes. For sensitivity. For assay optimization, the results highlighted the highest efficiency of the assay by adding inner primer of 0.8 micromole and LB6 form of 0.6 micromole. For sensitivity evaluation, the minimal amount of the detected DNA reached 5 petagram, consistent with previous vitro lamp. In conclusion, with the result of the present work, we conclude that molecular identification techniques are of great necessity to ensure proper labeling of salmon products. More specifically, a vitro lamp and floss lamp were developed in the present work with no races as little as 5 petagram of salmon salad DNA could be detected. Moreover, the self-constrained probe ACEs embraces a higher tolerance to impurity than non-specific dyes and can be used in the development of rapid diagnostic kit for salmon salad. Thank you very much for joining and happy for any suggestions. Thank you.